जय श्री राम दोस्तों। आज हम क्लास नाइन्थ का साइंस का चैप्टर नंबर 2 सेल द बिल्डिंग ब्लॉक ऑफ लाइफ के बारे में बातें करेंगे। आ जाइए ज़रा। अभी हम लोग जानते हैं दोस्तों कि हमारे पास सेल, देखो, ऑल लिविंग ऑर्गेनिजम्स आर मेड अप ऑफ सेल। सब किससे बने हुए हैं? सेल से। द सेल रिप्रेजेंट द बेसिक लेवल एट विच लाइफ एग्जिस्ट। Some organisms, such as bacteria or yeast, consist of only one cell and are called unicellular, while others, like plants, fish, birds, or humans, are made up of millions of cells, multicellular, that work together. A group of similar cells performing similar a group of cells performing similar functions form tissues. Different tissues are organized to form an organ, and several organs work together to form organ systems, such as nasal pores, nasal cavity, trachea, and lungs form respiratory system. Okay, next. E- so, even when cells are organized into tissues, organs, and organ system, the cell remains the fundamental unit of structure and function in all living organisms. This makes us wonder how such tiny cells perform so many different activities. What are the different components of cell? How do cell in our body communicate with each others? Do cell live forever or do they die? In this chapter, we will explore the answers to these questions as we enter the fascinating world of cells. Let's study 2.1. What do we call the ability of the human eye to see two objects very close as separate and distinct? Imagine two tiny dots drawn on a piece of paper. As the dots are moved closer, there comes a point at which they can no longer be seen as separate. When viewed from the 25 centimeter, that is called the near point of human eye, 2 points separated by 0.1 mm can be seen as a distinct. Otherwise, they appear as a single point. This is called the limit of resolution of the human eye, which is 0.1 mm. A cell is usually too small to be seen by the unaided eye. This raises an important question. How have cell biologists studied the structure and function of cells that are much smaller than the limit of the resolution of the human eye? You have learned about the convex lens. Convex lens, or a combination of lenses, that is an objective lens and an eyepiece, are used for the magnification of an object to make it appear larger. Robert Hooke was the first person to observe a cell in 1665 using a self-designed microscope capable of about 2 to 200 to 300x magnification. While examining a thin slice of a cork, he observed a small box-like compartments and named them cells. In school laboratories, light microscopes are used to observe objects using different objective lenses. To achieve better magnification and resolution under visible light, explore different parts of microscope in your school laboratory and use it to observe the fine structures of various materials. Now, you can see there's a beautiful design of the microscope. It's a light microscope which is also used by the Robert Hooke to look at the cork of a thin slice from a tree, and he observed that bark and called compartment. Next, we have the size unit conversion. 1 millimeter is equals to 1000 micrometer. 1000 micrometer. I hope you understand how small the size of the cell is. Estimated size of the onion peel. So there is a experimentation on the onion peel which you will study in your classroom laboratory examples. Next, we have a structure of a cell. So we have a structure of a cell. You have learned that the cells are organized into specialized tissues and organs and collectively perform specific functions. For these cells to function as units, they must be able to interact with each one another and with their surroundings. These interactions occur at the cell boundary, where substances move between the cells and their external environment. Even single-celled organisms exchange materials and respond to the environment through cell membrane. Next, cell membrane, the universal feature of a cell. The cell membrane is a thin boundary that surrounds a cell and protects its contents. It defines the individuality of a cell and is also called the plasma membrane. The cell membrane is selectively permeable, which means it allows some substance to pass through it while blocking others. You have learned about how oxygen and carbon dioxide move across the membranes of the alveoli in the lungs. How does the structure of a cell membrane in the cells of alveoli controls the movement of substance across it? Now, cell membrane, let's move to the other experimentation. This is our experimentation. And now, we have a thing that is called osmosis. That is called osmosis. So what happens in the osmosis? The movement of water through a selectively permeable membrane is called osmosis. The movement of water through a selectively permeable membrane is called osmosis. So what actually happened? And there is another thing that is called, when there is a difference of concentration, difference in their concentration, then this osmosis happens. Now there is another word that is called diffusion. Diffusion is the net movement of particles from a higher to a lower concentration, which occurs even without a membrane. Osmosis is the diffusion of water across a selectively permeable membrane. In plants, water from soil enters the root cell by this process of osmosis. I hope you got this. Now, we have three types of solution: isotonic, hypotonic, and hypertonic. So, when solute concentration of extracellular, solute concentration of intracellular, isotonic solution, right? So, if you look at it, in this thing, in this experimentation, if you see, or in this experimentation, there is a swelling. You see, the water that was there, right? The water is absorbed by those small particles that is there in the water. So it becomes more, it swells. That is called hypotonic. When there is less water inside it and the water moves from lower concentration to higher concentration, or from higher concentration to lower concentration. Next, in hypertonic, these particles started to put water, right? So if you are seeing here, the water is moving out of those small particles, right? And they are shrinking. But in isotonic, there is no difference. Like the concentration of water is same. Here the concentration of water is outside is more as compared to the inside. So water moves from higher concentration to the lower concentration. In the second case, the concentration of water is in these particles are more. So water moves from higher concentration to lower concentration. So this is about our cell, how small is the cell, what is the size of the cell, and what the cell performs, and what is different types of solutions on the basis of concentration of cell, right? So the membrane is a lipid bilayer layer of special flat molecules which water attracting heads towards out and water repelling tails inwards when the protein embedded in them. So listen what actually happens here. So structure of a cell membrane. So there is a protein. So these are the proteins and they are responsible for the water. They heads outward and water repelling tails inwards. So there are water repelling tails that puts the water inwards and some puts the water outwards. Next. So the molecules can move sideways, flip and rotate within the membrane, hence it is fluid. So the molecules can move sideways, flip and rotate within the membrane because if it is fluid, fluid can behave the way it wants. I hope, right? Proteins in the membrane acts as a gatekeeper in helping substance pass through. So these are the proteins that help as a gatekeeper. Next, we have an activity in the later part of this. So it is called tiles in mosaic. It is called mosaic model. Why? The molecules are arranged like tiles in a mosaic. It is called mosaic model. So it's kind of a model where we see how they are arranged in the form of tile. Like if you look at this. This is a clear example of cell in a onion, and you see a beautiful structure has been provided, right? So I hope you understood about it, and I don't want to make this video a little longer, so I just have to finish the introduction soon. So हमने introduction पढ़ा. We study about cell, unicellular, multicellular, what are the cell membrane, osmosis, hypertonic, and hypertonic, and so size of cell. So we will continue discussing about cells in our later classes as well. So this was just introduction. Till then, you enjoy and God bless you. Take care of yourself. Om Namah Shivaya. Jai Shri Ram.